This is the recording for the last two sections of chapter 21. 21.6 is on the supergroup Amoebozoa. Amoebozoans are protozoans that move by pseudopods. Now, we did cover the Rosaria, which also have pseudopods, but the difference is their pseudopods are more like thread-like or needle-like. And you'll see a picture in a minute. The pseudopods of the amoeba are just different. They're not as skinny. They're fatter. But the amoebozoa also do not have a test or shell. So the amoeboids move and ingest their food with pseudopods. So they move and feed with their pseudopods. Let's look at a picture. So if this is a typical amoeba, which has it's shaped kind of like a blob, you'll notice that it has these extensions of its cytoplasm that sort of stick out, and that's a pseudopod. Here's another pseudopod. And if you watch them while they move, those pseudopods change. Their shape changes constantly. So that's what I mean by pseudopods, and those pseudopods are used for feeding, and they're kind of used like arms. They reach out and grab the food, and then they engulf their food by phagocytosis, which means that they take the food in and form a vesicle around the food. And here's what a food vacuole looks like. There are a lot of them inside of this amoeba. But the other thing is that they move with their pseudopods. And we'll see that in lab. We'll get to watch them move. They feed, again, by phagocytosis, which is a form of endocytosis, bringing particles in through a vesicle that pinches off from the plasma membrane. Amoeba proteus is commonly studied in biology labs, and we're about to study an amoeba proteus tomorrow. Entamoeba histolytica is a parasite of the human colon. You may have heard of the disease called dysentery. You especially hear about that disease in cases of during wartime, when you have troops that are living large members of military are living, outdoors, and they're in the middle of a war fighting on the battlefield, that kind of thing. Dysentery has been common in the past. Naegleria fowleri is known as the brain-eating amoeba. It can cause a fatal brain infection. It lives in warm fresh water, and it can be acquired by inhaling water when swimming. So I know there may be some of you that have done this, but if you have sinuses, you may have heard or used something called a neti pot. And you have to be very, very careful, because what you do with it literally looks like a little teapot, and you pour it into one nostril, and then the water inside of it comes out the other nostril. But the danger of that is that if you're not careful and your water is not sterile, if you do get one of these amoeba that get into your nose, that it can actually end up in your brain. It is rare, but it does happen. So you have to be really, really careful about sterilizing and washing your neti pot. And you have to be careful about when you're swimming, about letting water get in your nose. But it still is very, very uncommon. So I don't want you to hear this and get scared. It may be something you want to read about. What are the risks of swimming and contracting Naegleria fowleri, brain-eating amoeba? It does happen. Now, I'm not talking about swimming in that's one of the reasons we use chlorine, I mean in freshwater lakes and ponds. Okay, now, in addition to amoeba, The supergroup Amoebozoa also includes the slime molds, and those are divided into plasmodial slime molds and cellular slime molds. Now, in chapter 22, we're going to talk about mold, true mold, that are found within the kingdom fungi. If they're the molds that are found, water molds and slime molds that are found within the kingdom Protista are not true fungi. They're not true members, or they're not, I guess you can say they're not true members of the kingdom fungi. So, but they are in the supergroup Amoebozoa. They are the plasmodial and cellular slime molds. And I'll just show you some pictures. These are plasmodial slime molds. And I don't see a picture of the cellular slime molds, but plasmodial and cellular slime molds and amoeba, the organism that we're going to study in lab, are found in the supergroup Amoebozoa. So now we're going to do the supergroup Epistheconta, which won't take long because there's just one member that we study. The other members of this supergroup are the animals and the fungi. But the protozoans that are found within this supergroup are the choanoflagellates, which are thought to be the ancestors of the animal called the sponge. These are some pictures of colonial choanoflagellates that live together in colonies. And we'll learn about the sponge later on when we get to our chapter on the invertebrates. So basically, just make sure for this chapter, chapter 21, that you go back and find that table. Know the name of all of your supergroups: Excavata, Chromalveolata, Rosaria, Archaeoplastida, Amoebozoa, Epistheconta. Make yourself some type of either flashcards or some type of organization to your notes to where you're able to test yourself. You need to memorize the name of the supergroup and the members of the supergroup. And then just focus on your chapter 21 practice test. Those questions are what you have to know. So you don't have to guess about it, but it is memorization that you need to do. It's not something that these are not names that you say in typical conversation. These are organisms that aren't common. We don't talk about them every day. Maybe the algae, but you probably never heard of a diplomonad until we just started this chapter. You may have never heard of an apicomplexan, even though I know you've heard of malaria, which is caused by an apicomplexan. But you just need to know this table, and then you need to make sure that you study that chapter 21 practice test. A lot of what's in that practice test are examples of these protozoans and algae. So that'll be it for this. This is a short little recording, less than 10 minutes. And so make sure you take notes. If you didn't take notes, go back and take notes. Copy down the notes that are highlighted in yellow. And we will start chapter 22 in class on Wednesday.